right, here we are back at these notes again, and we've already filled half of them out, right? We have DNA stands for deoxyribonucleic acid. There's 2 jobs of DNA. DNA replication, which is making an exact copy of DNA, and then protein synthesis, which is making proteins. And that's what we're focusing on right now. We learned the central dogma, which is DNA goes to RNA and then RNA to proteins. It's 2 steps, step 1 and step 2. 2 steps. So last week we learned about step 1. Transcription takes place in the nucleus. Helicase unzips DNA. RNA polymerase adds RNA bases. We have a new base pairing rule, A with U. And then lastly, the mRNA is going to leave the nucleus and enter the cytoplasm. So that's kind of where we are starting now. So step 2 is called translation. Write this again so you can see it better. And translation takes place in the cytoplasm. And we have 3 different kinds of RNA. And this is where we're going to actually see all 3 different kinds of RNA working together to make proteins. So before we just learned about mRNA, which is messenger RNA, but there are other types of RNA. So we have mRNA. It is going to bring the codon. And it brings those codons to the ribosome. Then there's tRNA, and what tRNA does is it transfers the amino acids. I'm just going to write AA because I don't have a lot of room here, but this stands for amino acids. It transfers the amino acids to the ribosome. rRNA, which is ribosomal RNA, it basically makes up the ribosome. A ribosome is made of RNA and some other proteins. And then lastly, the ribosome has a very special job. And it's going to bond these amino acids together. So if we look down here, this little picture right here, this big old, look like maybe a mushroom or something like that. This is actually the 2 parts of a ribosome. Okay, there's 2 subunits of a ribosome, and this is kind of what it looks like. Again, remember that we are in the cytoplasm. Step 1. mRNA brings the codons to the ribosome. So here we go. First codon, A, U, G. That's our first codon. Second codon, G, U, G. Third codon is A, U, U. We're just copying them over from over here. We're just taking them straight over. Now down here, we have this little tRNA, and they kind of look like this little T shape. And that's where the name comes from. On one end, they have an amino acid, and on the other end, they have something special called an anticodon. These anticodons match up with the codons, and they bring over the amino acid, and then we get a list of amino acids in the order that they need to be in. So how we find what amino acid is going to be brought over is we need to use our amino acid table. So I gave y'all two different kinds of amino acid table because some students like to use the circular one and some students like to use the square one, and you won't really know which one you like to use until you've tried both of them. What we do is we look at the mRNA codon. We read them in sets of 3. So our first mRNA codon, AUG, 3. So what I do with my circle one is I start in the middle and I work my way outwards. So I'm going to first find A, then I'm going to work out a little bit to U, and then I'm going to work out a little bit again to G. And when I do that, I find that I landed on methionine. But for amino acids, we only write the first 3 letters. So we're just going to write met right there. Underneath AUG, down here, I'm going to write MET because that is the first amino acid that I'm going to get in my line. Now I'm going to do the same thing again, AUG, but this time I'm going to use my square amino acid table to find it. So first, I look at the first letter right here, then the second letter at the top, and the third letter on the side, and that's going to move me into these different boxes, okay? So first I start with A, I'm in this row, okay? U, now I'm in this row, so I'm in this box right here. And then G is going to tell me I'm at the bottom right here, AUG, which again is going to give me a met. So no matter which table you're using, you should always get the same answer. So our first amino acid is met. Now let's look at our second mRNA codon. Our second mRNA codon is GUG. So I'm going to look over at my circular amino acid table, start with the G, move out to the U, and move out to the G again. And I see here I have valine. And again, I'm just going to write the first 3 letters, which is VAL. So here I have val. And again, if I did it on my square one, I would find the same answer. So here we have G, U, G. Again, val is going to be my answer for that one. So it doesn't matter which one you use. And lastly, AUU. AUU is going to give us ILE. So that's going to be our last one. ILE. Before we can call this amino acid sequence a protein, a very special thing needs to happen to this amino acid sequence, and that is that it is then folded into a protein. That's the very last step. Folded into a protein. All right, so let's do a little bit of practice. So typically when we do DNA to RNA and then RNA to a sequence of amino acids, which is then folded into a protein, we are usually given a DNA template first. So typically that is the information that you start with, the DNA template. So that's what I'm going to go ahead and write down right now. Okay, so here I have my DNA template. This is typically what's given to me first, and then I can go from here. So what I want to do with this DNA is I want to do my matching up. But remember, instead of writing down T's, I'm going to write down U's. So in my mRNA strand, I should have 0 T's, and I should only have U's. So I do my matching. So T matches with A matches with U, and C matches with G. T matches with A, G with C, A with U, C with G, T with A, C with G, C with G, G with C, A with U, A with U, C with G, and T with A. So notice that my mRNA strand is completely different than my DNA strand. My DNA strand has T's in it. My mRNA strand has U's in it. No T's. Now what I'm going to do is use my mRNA codons. And remember that I read codons in groups of 3. So this will be my first group. 2, 3, 4, 5 codons here. I'm going to use my amino acid table to find what amino acid these mRNA codons code for. I'm going to be looking at my square one. And I'm going to start out with AUG. And here I find AUG right there. And again, that's going to be a MET. MET is a very special amino acid because this is an amino acid that is called a start codon. It is always going to be AUG in mRNA and code for a MET. Every time. You always start with AUG. Always. That's our start, no matter what. Then we're going to move on to our next sequence of amino acids. I really want you to practice at home using your amino acid table to find the amino acids. Pause the video for a minute, try it, and then come back and check your answers. So now I have MET, thir, glue, ala, stop. Now, the thing about the stop codon is that it is also special in the way that this is not an amino acid. It just tells us it's time to stop. So when you see a stop, you need to stop. And that does not count as an amino acid. So if I ask the question, how many amino acids long is this amino acid sequence? You would say it's 1, 2, 3, 4 amino acids long because the stop does not count as an amino acid. Okay, and I also wanted to talk to you about some of the differences between DNA and RNA is that they start with a different letter. But the reason why they start with a different letter is because they actually use a different sugar. So remember that DNA is deoxyribose sugar. But RNA is not a deoxyribose sugar. It is just a ribose sugar. So that is one difference. Another difference between DNA and RNA is that DNA has the matching of A with T. But with RNA, we know that A matches with U. Right? And then another big difference between DNA and RNA is that DNA is a double helix, which means it's two-stranded. Where RNA is just single-stranded, it's just one strand. Okay, those are some of the big differences between DNA and RNA, and I just wanted you to be aware of those.